Thank you. So what I will do is, first of all, I want to thank you, and I think this is a really, as I'll come to towards the end of my talk, bridging countries, especially this bridging India and the United States, is very important if we're going to ultimately have success in the rare disease space. It's more so in the rare disease space than in many other spaces, because we're not going to be able to have commercial viability for many of these products unless we have a larger number of individuals that are eligible for them. And by essentially pooling our resources for development and then pooling the patient numbers, it may make a big difference. So what I'm going to talk about today, I'll talk about some of the challenges in product development for rare diseases, talk about options for the development of therapeutics. I'll try to convince you that gene therapies actually make a fair amount of sense for rare diseases if you're not already convinced, and talk about some of the things we're doing at the FDA to try to move things forward. So rare diseases are challenging, and they're challenging because the natural history of the diseases can be very limited. For some of these, we have very limited natural history. And for some diseases, even the natural history that we do have is very variable, because the disease manifestations, either because of the underlying nature of the disease or because the disease host interactions, can be very diverse. Especially the neurodegenerative diseases, the time period over which things happen can be very prolonged. That's challenging for us as drug developers, because we like clinical trials that last months to a year, not years to decades. And the other issue for the development of products is that in some cases, we're not looking, because it's impossible, we're not looking to reverse decline. We're just looking to stabilize decline. And that may be very, very important in, for instance, the muscular dystrophies. But again, very challenging to measure endpoints. And then in some cases, it's hard to find biomarkers, although I'll try to convince you that if we work hard, we can try to get around that difficulty. So let's think about, how do you address rare diseases? Well, whoops. Let's see if I can, there we go. You have small molecules, protein therapeutics, antisense oligonucleotides, and gene therapies. And I just want to take you very quickly through a tour of what we can do with each of these. So for small molecules, we understand how to develop these very well in terms of the product development pathway. Generally, making them is relatively straightforward. The good news is, when you have adverse events, most of the time, not always, but most of the time, adverse events, when the drug washes out, the adverse event goes away. And pills and even conventional IV administration drugs, they're pretty easy to give when they're small molecules. Disadvantages: anything you need to take over and over again is a challenge. To be honest, even drugs that are practically curative, like the drugs for chronic myeloid leukemia, imatinib, if you need to take it over and over again, it can actually cause challenges, because people are not good at repeatedly taking medicines. Sometimes it's difficult to predict the adverse event profiles. Cost of R&D for small molecules is pretty high generally, because you're generally doing things like library screens, or even rational drug development can be pretty high costs, and they're not curative. Protein therapeutics share a lot of the same things, and I'll just highlight the fact that they just tend to be more expensive, and the cost of production that drives that expense is obviously higher, but they share the fact that repeated administration is necessary, and they're generally not curative. I'd say antisense oligonucleotides, special case where we understand how to make these now. They share a lot in some ways with small molecules, in that we understand there's a business model that works for them, but you need to give them over and over again. And they do share with small molecules the fact that sometimes they have unexpected adverse effects, which makes it a little bit challenging to go from antisense oligonucleotide to antisense oligonucleotide. And again, they're not curative, but they've made a big difference. And finally, there's gene therapies. And hopefully I can convince you that, the idea of if not a one and done cure, at least a therapy that you can give once and can have very prolonged effect lasting perhaps a decade or more, I think is a very attractive option. If we design the gene therapies right, they have a reasonably high probability of success just based on their design. We can go through many different diseases because if you can clone the gene, which since we have the whole human genome now, has been done, you can hopefully address the disease with one of the vectors that we have available. And this concept of long-term cure or your long-term treatment or cure is very attractive. Now, there are some real disadvantages, which we'll go through in the next little bit. They are pretty challenging to manufacture. There's complexity involved and pretty high cost relative to other pharmaceuticals. They do have the potential for irreversible side effects. Genotoxicity is a possibility, but hopefully we can mitigate that by doing our jobs correctly, and the benefit risk can be kept favorable. One does need to know what one is doing when one is administering these in order to minimize the chance for side effects. And at least in the United States, I can't speak for India, but at least in the United States, I suspect for India too, this is a very new business model because we're used to drugs that you have a revenue stream where, every pill is sold. It's part of a revenue stream. Here, it's one dose and you're done. And in some ways, it's a very challenging model because for some rare genetic diseases which don't arise because of spontaneous mutations, if you do your job well, you put yourself out of business in 5 to 10 years because you've treated everyone who needs to be treated, and then you just have small numbers to treat. So it's a challenge. Nonetheless, we now have 15 gene therapies approved in the United States. These include cell-based gene therapies. Those are the ones in bright blue. 2 stem cell-based therapies, Zynteglo and Skysona, one for thalassemia, the other for adrenoleukodystrophy. And the remainder of these in purple, various directly administered gene therapies ranging from a locally administered gene therapy for hereditary retinal disorder to gene therapies now for hemophilia A, B, and for Duchenne's muscular dystrophy. So really a nice portfolio of gene therapies, and it's going to increase over the next year. Just to say, what does gene therapy look like when things go really well? Well, I think anasemnogene, which is the gene therapy for spinal muscular atrophy type 1, is a great example of what things look like when they go really well. I don't think we're going to have to expect things to go this well, right? We said stabilization was okay, but when things go really well, you have a gene therapy for a disorder which normally leads to essentially inability to control motor function and death by age 3 to a condition where if the gene therapy is given within the first 6 months of life, the children develop normally. They show no signs of SMA. And on the right-hand side of this slide, you see a child running around at age 3. I don't mean to try to put statisticians out of business, but you don't need statisticians for this, right? If you have, 14 out of 15 children running around like that when they should be dead, it's pretty apparent. So this is what happens when things really work well. The problem is there are some challenges here. And I think in the United States right now, we are at a place where we're really at a critical juncture where I think we can either see an acceleration of the pace in which we have new gene therapies come to market, or we could have a stagnation. And I don't mean to essentially oversell what we do at FDA, but I think we have a significant role to play here. But the 3 things I'll tell you about today is we have manufacturing challenges to deal with. clinical development timelines, which I think have put challenges and have made some sour, at least in the investment community, on gene therapy, and different global regulatory requirements, which I think is a great thing to be talking about today to this group. So let me move on here. So at the Center for Biologics, we're trying to deal with all of these issues. Some we have more under our control, I'll admit, and others we have not so much under our control. We're trying to help advance manufacturing technologies for cell and gene therapy through research. We actually have small grant programs, and we have in-house laboratories that are working in this area. I'll tell you a little bit about those. We're going to work to try to more clearly define the use of accelerated approval for gene therapy. That's something that's very much within our control. I'll tell you about that. We're looking to see if we can get towards better global regulatory harmonization, because there again, we think that may really help this whole field by creating markets that are where we might have only 50 people in the United States with a disease. There'll be 100 in India or 200 in India. You go ahead and look at Europe, et cetera. You suddenly have markets that are actually of a size that a sponsor would be more interested. And then finally, building on what Frank already mentioned, we'll talk about a pilot that we're initiating based on what happened in Operation Warp Speed. So just to start with manufacturing, our problem is that right now, the profitability of gene therapy is when you have somewhere between 100 and 200 treatments per year. Why? Because that's where the capital investment in manufacturing pays off with what one can charge for the gene therapy. And so the sweet spot is the medium-sized cup of coffee here. For another day, probably 5 or 10 years from now, if you ever invite me back, which may not happen after this talk, we'll talk about what you do for manufacturing, gene therapies for the masses. It's very challenging right now based on the current generation of adeno-associated and lentiviral vectors because of the way we have to make them. But the problem actually is the small cup here. And the reason why is because the cost of setting up and making each gene therapy is such that that's almost a fixed cost. And it's almost the same whether you're in the small cup or the medium cup. The problem is the small cup doesn't have with it the profitability right now of the medium-sized cup. And so what we're seeing are gene therapies for small populations falling out of development. And if anyone doesn't believe me, you can go and just read the financial papers about programs that have been either dropped from development, handed back to academics, or kind of on the auction block, because the larger companies have just found that they're not going to be profitable for them. And in fact, some larger companies have just dropped their rare disease gene therapy development programs entirely. But what can we do about this? Well, one of the things, I'm just going to, some of the things we can do is think about how we can make manufacturing more efficient. And one of the ways we can do that is by actually automation. In the chimeric antigen receptor T cell world, one of the ways that, for instance, China has found to try to decrease cost is to automate this. We can do it in the same thing. We have people in the United States automating this to try to bring down costs. Because especially if you have closed systems for production, you don't need the same type of large clean rooms to make these things. Again, decreasing capital costs, hopefully bringing down the cost of these products will help. We could do the same thing for gene therapies. It's just that nobody has spent the time doing this. They are working on this now in various labs, and hopefully within the next 5 or so years, we'll see not a soda machine, but essentially a gene therapy fabrication device, which will have actually kind of a nice business model of a device with disposables to make these therapies. So this is just one of several things. I mean, some of what we have to do in the United States too is kind of harmonize more in terms of the processes we use. We need to exchange more information among companies, but we'll leave that for some other time. And I want to move on to what we can do at FDA. One of the issues for us is that we have an accelerated approval pathway. Frank knows this all too well, which allows us to give things an initial approval if they're for a serious or life-threatening condition based on substantial evidence of effectiveness based on a marker, biomarker, or intermediate clinical endpoint that's reasonably likely to predict an effect on a clinical endpoint, like how someone feels, functions, or survives. It gives us a lot of leeway in what we can do. And then ultimately, we have an approval based on that biomarker, perhaps, or intermediate endpoint, but we have to come back with a confirmatory trial, which does show clinical benefit. Our statute doesn't say how fast one has to come back with this. So it's possible that for some of the rare diseases we're dealing with, it may take several years before one goes from an accelerated approval to a full or traditional approval, but that's okay. I don't think any of us, at least in the Center for Biologics, has much problem with that because we understand that's just the nature of the diseases we're dealing with. If we're going to leverage accelerated approval, though, gene therapies have this really nice property that allows us to leverage something, which is that in many cases, because of what we can do, we can develop animal models by knocking out a gene, or sometimes there are natural animal models of a disease that we can then administer gene therapy to and see if we bring up the level of a given protein to a certain extent, or we knock down the level of a harmful protein to a certain extent, do we reverse the disease process? So that's an animal, on the animal side. Alternatively, in some humans with different genetic diseases, there are people who have reduced levels of a protein, but who get along normally. And if you can figure out what level of protein expression correlates with normal, that's the criteria you need to be able to use accelerated approval, because then if you can show that you can use a gene therapy to get to that level, you have something that is reasonably likely to predict that you'll have a clinical benefit. So hopefully, by using a combination of animal models, human observations, one or the other or both, we'll be able to leverage using accelerated approval to get more therapies to patients. We're going to have to do this because more and more, because for so many of the rare diseases we're dealing with populations of, handfuls of patients where if we were to try to do randomized trials, they would be impossible for various reasons, either because we didn't have enough patients, we didn't have enough time, or because people just simply wouldn't be randomized. I want to move on to global cooperation, because that's clearly something that we can do together as regulators that could make a big difference. Prior to the pandemic, even prior to the pandemic, we were working with WHO to try to come to harmonization in the cell and gene therapy area. This has been a longstanding area of interest because many low and middle income countries could benefit tremendously from gene therapies. Supportive care requires a lot of infrastructure to administer. If you think of it for beta thalassemia major in the United States, look, if you have beta thalassemia major, I'm a hematologist by training, you can live practically a normal lifespan. You get blood transfusions every 2 to 3 weeks. You get iron chelation therapy. You get MRIs to make sure you're not getting iron overloaded. The cost of that is pretty significant, but it's not even the cost, it's the infrastructure required is very significant. On the other hand, a one and done gene therapy requires much less infrastructure over the long term. And so it's probably true that if we had a robust program for administration of gene therapies in various diseases, we could hopefully bring benefit to people, not just in high-income countries, but to more people in low and middle-income countries. But the only way we'll get there, I think, is if we make the process easier and get there with some regulatory harmonization, in part because we can't have difficult, especially for very small populations, it's difficult to go from one country to the next such that it costs a lot of money to reformat applications or to get over different regulatory requirements, we're not going to get there. So we're trying to get towards convergence of regulatory approaches in high-income countries with the eye of trying to then perhaps get something to, like, could it be look like pre-qualification for gene therapies, much like we have pre-qualification for vaccines with WHO. And so that's all something we're working on now. It's in early stages, but it's getting a lot of traction, and I hope to move that forward. And then there's this question of whether, aside from everything we could do with harmonization, could we expedite the review in different countries by pooling resources? So instead of having different countries review the same application separately, could we do what's being done already in the cancer medicine end of FDA, of having 8 or 10 countries come together and review the applications together, and when they come to a decision, each of the different regulators can then take that home and have an approval with much less effort put into the process. So that's the Project Orbis model, and we'll see if that can work in this area. And then finally, just to finish up, you know, what we learned during the pandemic, and it was really nice that you set this up, Frank. We learned how much exchange and dialogue between developers and regulators can facilitate product development. You know, although it might have been a little exaggeration that we didn't get too many calls at midnight. Although we did have an operation that people, you know, we were going pretty much close to 20 hours a day of back and forth with regulatory advice. And the idea was that we didn't want to have anything delayed because of questions that could be reasonably easily answered. And one of the things that came out of that was not only just answering questions, but recognizing that oftentimes questions generate questions, right? We give a response and someone says, hey, but what do you mean by go do the usual quality analyses at the usual time? What do you mean by that? And so you can say, oh, we want you to check sterility of the final product. And then they know what you're actually talking about. I'm just joking. I mean, what I'm trying to say here is that it allowed us to have this back and forth. And if you look at what happens in gene therapy development right now in the United States, oftentimes small companies ask us something, they wait 60 days to get a response. We answer something up with an answer that might not be super clear. They try to interpret it kind of like the Oracle at Delphi and read into what our comments are. They may make them, they either then ask another question, or they go off and do something wrong, and then they come back to us a couple months later and we say, no, we actually meant X. Or they ask us the question and then it takes us another 30 days to 60 days. So we draw things out. So this concept is, if we were able to have real-time discussions, could we bring rare disease gene therapy to the market much more quickly? Actually keep more products in development, because small companies don't have a quarter's worth of cash to burn just to wait for us to get back to them about a question from FDA. So this concept is a start pilot. It's a small pilot, but the idea here is to take a couple of products that look very promising in early clinical development, give them this warp speed service, you know, where basically we commit to having a project manager that they can contact. If they call the project manager, email the project manager, we'll get back to them very quickly, within a day, or if a meeting is necessary, within a few days, to have that meeting to help move development forward. And we've asked for applications to this. At this point, we published a Federal Register notice, which is our way of essentially like putting out an advertisement that we want proposals. And we'll be getting those in and choosing some of these. And then the idea is we'll measure. We know how long it takes to get from place to place. And we're going to also be asking the companies for their feedback. And we'll see if we can actually move things forward in a much more rapid manner. Now, I think we all have, I mean, I have my bias of where this is going to come out, but we have to prove it so that we can expand this because it will take a lot more resources if we do this on a regular basis. On the other hand, those resources may be met in spades by the benefits to patients. And that, I think, shouldn't be underestimated. So essentially, just to sum up here, we're very committed to working with the community in gene therapies for rare diseases. I think we very much look forward to collaborating with colleagues globally. I think India, an incredible opportunity given the fact that, you know, I mean, one of the wonderful things, I've worked with Indian partners since my first job in industry in 1999. I think I know that we have tremendous capacity and capabilities, and I think leveraging them together, hopefully we can do something very good for people with rare diseases. So I'll stop there. Thanks very much. Questions now?